Hi, everyone. Good afternoon. I want to welcome everyone to the Bird Genomics community meeting. My name is Mara. And if this is your first community meeting, welcome. So the point of these meetings is we want to have collaborators who use our data or folks who develop cool tools with our data present to the Broad Genomics community as well as the wider Broad. So if anyone has any ideas for topics you want to hear or different cool talks, probably the easiest thing to do is email genomics@broadinstitute.org, and then we will take your suggestion into account and hopefully give you guys what you want to see. So today we've got Sam Friedman here. He's from the Data Sciences platform, and he's going to give a really cool talk he gave at the LGBT meeting in February. And so despite the fact that there might be the presence of a lot of cats in his talk, apparently, sadly, he's allergic to cats. So, but anyway, I just want to welcome Sam, and I think he's going to do questions at the end. So you guys hold your questions. There'll be time at the end, and I will be running back and forth with the microphone, helping you guys ask your questions. So thanks for everybody, and here's Sam.
Thank you, Maura. And there's some seats in the front row if people are craning their necks in the back. So we're going to talk about variant filtering and calling with convolutional neural nets. And to place this in context, we'll talk a little bit about the history of variant filtering. So I'd like to say it's always handcrafted. It's just what are the filters handcrafting? So in the very beginning, we handcrafted the decision boundary. So where we said a good variant existed and a bad variant existed was this very hand-tuned line that different analysts would draw based on the data set and their own expert knowledge of the domain. The problem with that is as it doesn't really scale well, because when you get a new data set, sometimes your good variants look a little bit different, and those handcrafted, finely tuned boundaries need to be adjusted. So the next step is to learn with a machine that decision boundary in a sort of automated way. And the way you do that is you, instead of handcrafting the actual decision boundary, you handcraft features, which is certain ways of describing your variant, which should be more robust to different data set generation. And then the machine can learn the boundary in this feature space or annotation space, and it will draw the line there. The problem with that is these handcrafted features are all the things that humans have decided are important to determining a good variant from a bad variant. But there may be things that we've missed in our expert studies. There may be small patterns that are very hard to pick up by a human that are actually available in the data and could be helpful for classifying our variants as good or bad. So to address that, we start learning the features themselves. So now we're not just at that last layer of deciding whether a variant is good or bad, but the actual features that get extracted from our data are learned in an automatic way that we will explore throughout this talk. And this all foreshadows some future day where we learn the architectures themselves in a machine and we can all retire. And at the left, you see Frank Rosenblatt, who is the inventor of the first sort of learned artificial neural network called the multi-layer perceptron. And it's perfect because you see him actually hand-tuning the wires. And back in the day, that was like him twisting these potentiometers and little transistors that could actually get bugs stuck in them. But you see it's always a sort of handcrafted process. It's just you sort of remove yourself, or you put layers between you and the data. So another sort of historical relic of this practice is something we called the Variant Review Lunch, which took place around the corner in Voyagers with lots of pizza and lively arguments about what was a good variant and what was a bad variant. We would load them up in IGV and hash it out, and if we all agreed, we would add a variant to our human annotated database of good and bad variation. So ultimately, it was the humans who was deciding what the truth was back then. how did the humans do it? Well, we have these neural nets we carry around on our neck, and they have these sensitive, gooey spheres in the front that absorb light and turn it into electrochemical signals, which they can use to process IGV screenshots. And how do we know about that architecture? So here's a little video, it's just a minute long, about how some of that architecture was elucidated.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen, and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen. And we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina. And every time we did that, we'd get a response.
Okay, so there's a lot in that little snippet that we're going to dive deeper into throughout this talk. One, I just love that it was an accident, that like they were trying to activate this neuron, and that buzzing you hear is the neuron firing. So I love that it's them actually picking the slide off the projector that causes it to fire. And what is the picking the slide off the projector casts this line across the visual field of the cat, and it's an oriented line. So it's not that circle that they thought, like maybe there's a circle neuron that fires every time it sees a circle. But it's not that. It's the line, the oriented line, that moves across the visual field, which causes the cat's neuron to fire. So what does that neuron look like? It's in this mess of other neurons. It's an immensely complicated neural structure that has these beautiful drawings and has both sort of analog and digital components. There's this electrochemical gradient, but then there's also a discharge across the neuron. But we can abstract that away and sort of draw it like this cartoon GIF with just some little neurotransmitter squirting out, and it causes a neuron to fire. And we can extract it even further, abstract it even further, and just look at it as a weighted sum. So that neurotransmitter, those little red dots that are squirting out from one end of the neuron, are just numbers. And a neuron sensitivity is just a weight that responds to that number, that multiplies that number. And then whether the neuron fires or not is just summing those numbers together, summing those different sensitivities together, and then applying a decision function. That is, if the weights add up to a certain number, I'll fire. If they are smaller than that number, I won't fire. And beautifully and incredibly elegantly, we can describe this whole network. Now, not just one neuron, but up at the circled in red, with just, I think, 15 characters, a tenth of a tweet, less than a tenth of a tweet, you're describing a whole neural network. So AL is the activation of one layer, equals, and then we have a little sigma there, that's for the activation function. Critically, this has to be a nonlinear function. This can't be something that just multiplies and adds. It has to apply some kind of nonlinearity. It can be just a very sort of bent little hinge of a function, but it needs to be nonlinear so that you get some advantage from adding more layers. And then we have WL, which is the sensitivities, the weights at that layer, times A of L minus 1. So that's where we're talking about this hierarchy. Those were the activations at the previous layer. Plus BL, which is just a bias, which means we don't have to go through the origin. We don't restrict our lines to going through the 0, 0 point, but we can shift them. So just in those few characters, we're abstracting a whole neural network. We've lost a lot in this journey. We now are only talking about a feed-forward network. We've lost all the sort of intricacies of the frequency at which the neuron fires. The neuron doesn't just fire or not fire, but it has a frequency associated with that firing. And we've abstracted that away to just a number that is the strength of the neuron's firing. So a lot is lost, but a lot is gained too in this very elegant description of a neural net. And this journey of abstraction, this U-shaped journey of abstraction that we take, reminds me of this other journey of abstraction that Picasso took with this bull, where we start with a very sort of representational, accurate in some ways, depiction, and then we go through this process of both sort of pulling out and exaggerating the certain details that are characteristic of the bull, sort of in a way making the bull more of a bull. And then as we continue on this journey of abstraction, ripping away the extraneous till you get to this very, very compressed, very refined image. I mean, almost comically simple, but yet deeply expressive about what the bull is. And there's this quote, which actually isn't Picasso's, but is often misattributed to him, which is that good artists copy and great artists steal. And it's lovely that it's often misattributed to him, and it's because of Steve Jobs, I think, that it's so commonly thought that he said it. It was actually T.S. Eliot, but that's besides the point. What's really beautiful about that quote is that you can take something and use it, and you don't have to worry about all the details. And I think that's what neural nets, artificial neural nets, have done with the real neural nets. That we treated them as great artists treat them. We stole them. We're not trying to replicate what a real neuron does, but we are taking inspiration and running with it. And it's sort of like this tension between biomimicry and invention. While birds certainly inspired us to build flying machines, airplanes don't flap their wings, and that's okay. No one's upset about that. So we can be flexible. We can be happy with artificial neural nets and biological ones as well. So that cat got excited. That neuron in that cat got excited when it saw this oriented edge, this edge at a certain angle. And funnily enough, we already knew that was an interesting thing to look for. And we had a whole set of language to describe that. So what you're seeing now is 2D convolution over this beautiful drawing of a horse and the groom. And the box that surrounds the mouse is the output of the convolution. And what you're seeing now is a vertical edge detector. So you see the vertical lines pop out, and the actual numbers that describe the edge detector are shown in gray. There you're seeing diagonal edge detectors, and this is something called the sombrero edge detector, the Mexican wavelet, Mexican hat wavelet, because it looks sort of like a sombrero, and it detects edges in all orientations. Okay, and these were known in the computer vision community as good tools for various image processing tasks. And lots of computer vision tasks begin with some kind of convolution with one of these known filters. And they're called edge detectors. But they also have, in general, these properties of frequency selection, which is they pull out certain frequencies in the input and suppress everything else. And they're also translation equivariant, which is that as you move something in the input image, you will also move something in its response. And that encapsulates this idea that wherever the horse's head shows up in the drawing, it's still a horse's head. Its meaning hasn't changed because we've moved it around. So we want our representation to have that invariance, that the horse's head is still a horse's head if it's all the way on the left of the image. We can also convolve over 1D sequences. So here we're seeing a 1D signal in the GIF, but of course the DNA sequence itself is a 1D signal of different nucleotides, different bases. And here very scratchily, I've drawn the convolution of this TC, which is a short tandem repeat detector, or motif detector, we sometimes call them, with this larger sequence, which is depicted in just color space. So there, blue is C, brown is G, red is T, and green is A. And you see the line under that is the response, the output. And every time there's a TC, you see these spikes. That's where the motif detector and the sequence agree, and you get this big response. And we can look for all kinds of things in our input sequence, not just short tandem repeats. We can look for homopolymers. We can analyze GC content. And that's what these neural nets learn to do. And yet again, this is something that was sort of well studied in the literature and handcrafted before we had neural nets. So before we learned these motif detectors automatically, we would make things called multiple sequence alignments, where we'd look at a little sequence of interest and align it across different species or different homologs, and we could encode the sort of importance, the relative importance of each base at each position in something called a position weight matrix. And you see it sort of being built here in this slide, where we just sum up the different bases we see at each position, and that gives us that matrix of counts. Then we can divide that matrix of counts by the total number of positions we've seen, and that gives us a position frequency matrix, how often we see each base at each position. And then we can sort of have some kind of background guess, and sort of the most naive but not ridiculous guess is that we could have seen any base at that position equally. And then by taking log odds ratio, and this is sort of the end of the math that we're going to cover here, we can encode our surprise at seeing a base in a given position. So the null hypothesis is that all the bases are equally distributed, and then the alternative hypothesis is that they're not. And this position weight matrix encodes that that surprise, or that belief about seeing certain bases at certain positions. And that is what convolution is, too. So we talked about one being 1D convolution and the images being 2D convolution. And I think it's really easy to get lost in the dimension sauce. And I encourage you to just not worry about it. In a previous MIA talk, we plotted dogs and cats in 2D space. And so each dog and cat became a point in a 2D space where the two axes were weight and domestication, or ferocity. And we sort of cranked through that whole example, and you're welcome to watch it on YouTube. And we just talked about how the reference sequence, DNA, is a 1D sequence. But we can equally encode it as a 2D matrix, where we have one axis for the 4 different bases. So they're just 4 different, and I'm showing them as colors here. And then another axis for each different position in the base. So then it kind of becomes a 2D thing. Likewise with color, we often think of it as 3D somehow, that there's red, green, and blue, or hue, saturation, value. But you can also encode color as red, green, blue, and alpha, or you can have a 36 color channel representation for color. And I once asked on Quora, how many dimensions is color? And the answer was It's an infinite dimensional point in an infinite dimensional space, and I don't understand that at all. But it's just sort of, you pick a dimension, and there's a sort of a set of assumptions, but don't get too tied up with that number. That number is not set in stone. Likewise, for rotations, 3D rotations, you can encode those as 3 numbers. That is your rotation around x, y, and z axes. But you can also encode them with quaternions, which are 4 numbers. And that's a little bit of a better encoding. You get away with away from some problems that come with the 3D encoding. So all of these things, it's not such a clear-cut thing about 2D or 3D. Likewise, to the GPU, which does a lot of the learning that these neural nets achieve, it's what the GPU sees is actually a 4-dimensional matrix because it looks at many examples at one time. And then on the right, what we're seeing are these read tensors, which is a 3D representation of a variant where we have not just the reference sequence, which is that 2D thing, but also a dimension to store all of the different reads. So that gives us 3 dimensions of data about each variant. So just exploring more deeply this reference implementation, we can go from the bases to this 2D matrix. And also what's nice about this is we don't have to, this is called the one-hot encoding, but we can also encode some doubt about what the base might be. So we, along with the bases, get a base quality, which tells us how confident the sequencer was in the given base at that given position. And we can very naturally encode that into this matrix by distributing the weights across the different bases. Here is this 3D read tensor depiction of, anyone know what variant we're looking at here? Oh, I lost my mouse. But it's circled in pink. It's a, anyone? Yell it out. It's green, right. Okay, it's green because it's an A for half of them, but only half of them are an A. The other half are a G. It's a SNP. It's a SNP. It's a heterozygous SNP, a G to A transition. Or is that transversion? Here we have a, is this heterozygous or homozygous? Het. Yeah, it's a het because we see, again, a separation. Half of the reads have this extra extra TCTT, and the other half don't. So this is a heterozygous insertion. And here we have a homozygous deletion because they all had that same deletion. So this is the 3D object that we can convolve over, even though we just call it 2D convolution. Okay, so that's what our input is. What is our output? What do we want to say? Well, for the filtering task, we want to say, yes, this is a variant, or no, this is an artifact. How do we know? Well, originally we had these human-crafted from variant review lunch truth sets where humans had sat around and looked at a variant and decided it was good, and then looked at another one and decided it was bad. That didn't really scale that well. We weren't able to get so many known variants using that methodology, so we've developed others. One of the first was to just run a bunch of different variant callers. So run GATK and Strelka and deep variant and freebase, every variant caller you can think of, and then where they all agreed there was a variant, that became a true variant. Where there was some disagreement, that became unknown, and where they all said there was no variation, that became known reference site. So that's called like a consensus call truth set. And that's how the genomes in a bottle truth set works. The Illumina folks included also pedigree information. So not only did they have consensus calls, but they looked at Mendelian violations and also removed those from the truth set. So those are two very valuable truth sets we have. The trouble with both of those is because they're based on consensus, they sort of bias us towards the easy places in the genome to call. They don't look at the places where there was discordance. They just exclude those from the truth set. So they sort of narrow our vision. And to address that problem, some folks here at the Broad came up with this really fascinating idea, which was to take a complete hydidiform mole, which is a pregnancy, or it starts as a pregnancy, but the egg has no DNA. And the sperm comes and gives its DNA, and the first thing that happens is the sperm's DNA is duplicated. So it looks diploid, except the diploid, the two strains came from the same person. So it's really haploid. The two strains are the same. It's not viable, but it grows and it creates a fair amount of tissue, which can be sequenced. So people, I think at Washington University, sequenced this on long-read machines at PacBio, and then GP sequenced this many times for us on Illumina machines. And what's so valuable about this is because we used long reads and because it was haploid, we're able to look into parts of the genome that are very hard to call with only short read data. So from this, we were able to take, and we took two different CHM strains and mixed them together as a synthetic diploid. So we're able to look into parts of the genome that are much harder to see if you're only getting your truth from consensus calling. And I think it's a really beautiful sort of unique thing about the Broad is that we're able to take this very maybe tragic experience of having this molar pregnancy and turn it into a resource which we're freely sharing with the world that's really valuable. It's like this really weird thing that biology does that's also kind of sad and tragic, and we're taking it and making it into a freely available resource for improving variant calling. I think that's pretty cool, and a lot of GP folks have been involved in sequencing it, so great work. So that gives us some truth data. Now back to the biological neural nets where we got our original truth data. How did we get that? So we saw the first sort of gooey light sensors, spheres that these animals have that process the light, but that turns it into electrochemical stimulus, but then it goes into this deep neural network, the visual cortex, which processes it hierarchically. So it looks not just at those oriented edges. That's the first thing the neural net looks at. But then it looks for more complex features, more abstraction, layers of abstraction as we build up into the visual cortex. So when we design our artificial neural networks, just like great artists, we're stealing it. We're stealing that idea of the visual cortex. We're building these abstract layers of hierarchy where we get more and more abstract representations of the variant as we go deeper. So here we're seeing a bunch of different neural net architectures that we've experimented with. Some only see the reference sequence. Those are the 1D CNNs. We also integrate our annotation information, so that handcrafted set of numbers that describe the variant, we can also feed that to our neural net. And then it goes through these layers of abstraction and convolution, and then it comes out with a decision at the very end. This is the same idea behind deep variant, which used a neural net called Inception, which was designed to classify images, 2D color images, and classify them as one of 1,000 classes, many different types of dog, lots of cats, and airplanes, different things from the natural world. And so what they did was they ripped out that last layer of the neural net and replaced it with just 3 neurons, which stood for the genotypes, heterozygous, homozygous, or homozygous reference. So the deep variant architecture is really a genotyper. And what it takes in, because they're using just a neural net that was designed to take in images, RGB images, it takes in an RGB image. And the RGB image is essentially a IGV screenshot. So it's just 3 channels of information that try to encode the variant site. So we have a similar approach, except we're not ever reducing our representation to a 3D image. We're keeping it in this expanded representation where each base has its own channel. So we don't smash the bases together. And there's some other slight architectural differences in how we process this. We're careful not to treat our data like an image that has these properties of smoothness. Most neighboring pixels are similar to each other. And so because of that, you can design your network to sort of skip certain neurons, certain pixels, because you expect them to be the same. But we don't do that because we know our DNA data is very much discrete. And while there are homopolymers, most neighboring bases are not the same. Also, since we're designing a filter, we don't emit a genotype. We emit whether we think it's a true variant or a false variant. All the same, because we score our variants, we can compare to deep variant. And we're at least competitive with their outputs. These are results on a small test set of variants that haven't been seen by the neural net during training. And we also compare to the VQSR, which was our variant quality score recalibrator, which uses a sort of handcrafted features and machine learned decision boundary approach called the Gaussian mixture model. Here are some more results, and we're happy about that. So what do we call this thing? Well, we thought about calling it Marduk, which was this Babylonian dragon net hunter, or Neurovar, which is this like dubious neurostimulant that you could buy on Amazon for a while until it got outlawed, or a colander, which I really like because it like filters noodles, or Clarendon, the Instagram filter that makes your pictures all look the same. But we're calling it CNN score variance because the GATK philosophy is call the tool what it does. This tool scores variance. We're calling it CNN score variance. Okay, so people often complain about neural nets that they're just a black box, and yeah, you get a good answer out of it, but who knows what's going on inside there. But we know they're wrong because we can look in that black box and we can see what's in there, and it's weird, it's beautiful, it's strange. Let's take a look. At that first layer, what do we see? Well, it's exactly what that cat was getting so excited about. These are edge detectors. These are oriented edge detectors. And this is from a neural net trained on images, initialized with random numbers. And it's learning the same thing that evolution programmed into that cat brain. So to me, this is a really amazing feat that we took this architecture and we ran with it. We totally twisted it from what biology was doing, and we made it fit onto a computer, and we made it trainable on a GPU. And yet, after training it, after seeing millions of images, what pops up? The same exact structures that are in cat brains and our brains. These edge detectors, looking for edges. That's what that upper left corner, that's what it's all about. You can see there's sort of stripes at different angles. So every time one of those edges appears in an image, that neuron will get excited. But remember, the neural net is hierarchical. It has different layers. This is only the first layer. That's what Hubel and Weiss connected to. They connected to a neuron in the cat's first layer, the V1 of its visual cortex. But we can go deeper. What do we see at the next layer, at Conv2? Well, here, actually, if you look in the fourth column and the bottom row, you see a sombrero. You see it's dark, and then it's light. It's like whoosh. That's the Mexican hat wavelet. That's another sort of complicated, now it's looking for edges, but it's looking for edges in all directions. It's not only looking at one orientation, it's looking at edges all around. And it's doing some smoothing at the same time. So it sort of suppresses noise. It's a really beautiful convolutional kernel, and it comes in here just naturally from the process. Starting from random initialization and just tweaking the weights little by little, we arrive at that same structure that mathematicians had derived years before and that evolution had derived. So this incredible convergence happens in these features that the neural net learns. We go deeper, and we start seeing real texture being detected in Conv3. Things like paws and little segmented patterns and sort of furriness. A lot of dogs and cats in this image data set. So that's what the neural net learned about. And then as you go even deeper, you start seeing these part detectors like a duckbill or a paw or a dog nose. And what happens as we go even deeper, all the way to the very end, the very last layer of the neural net? What do we see? We see these things. And now it's a game of guess that neuron. So all of these are animals. Feel free to shout out if you think you see one. Which one? Penguin? Penguin, for sure. Anything else? Toucan? Millipede? Yeah. All right, one more round. Any more guesses? Yeah, so this is a lot of fun, and the way these images are made is you take this neural net that's been trained, and you use the same exact machinery that trained the neural net, but instead of adjusting the weights in the neural net, we adjust the image pixels. So we adjust the pixels to excite a particular neuron. And what's really cool about the packages like TensorFlow and Theano is that they allow you to describe this computational graph, which is the neural net, and then to take derivatives of arbitrary points in the neural net. So we use the same exact machinery, but rather than changing the weights in the neural net, we change the weights, the pixels, and we arrive at these weird and strange images that really tell us something deep about what the neural net learned. We can play the same game with our read tensors and our variant classifying neural net to pull out the features that this neural net thinks are important. And we're sort of just getting into this line of work, but we see a lot of familiar friends. We see the homopolymer detectors, like these triples. We also see a sort of het detector. Oh, I don't have a mouse, but top left is sort of a het detector. Remember how the hets separated into those two groups. We also see sort of deletion detectors or insertion detectors. And we can watch this process live, so we can look at what the kernels look like during training. So this is a neural net as it's being trained, a 1D neural net. So we're looking at the raw weights from the neural net during training, as it's becoming a classifier of variance. And you see things like the poly-T coming out, or what else? Get some STRs. Okay, here, we'll look at a poly-C detector being learned. You see, as the training happens, this kernel learns to pull out just poly-C. And it's actually suppressing it. So every time it sees a big row of C's in a row, then it gives a big negative output. But sometimes they're not so easy to interpret what these individual kernels are picking up. And an important thing to keep in mind when playing this game is that the neural net has many different kernels that it learns simultaneously. And it gets to use them all together to make its decision. So while looking at individual neurons is instructive, it's ultimately not the same way that the neural net is using these neurons. It's using them as an ensemble together. And this visualization shows the whole sort of suite of those first layer 1D convolutional kernels. And here it's much easier to see. You have like a poly-A detector, that big stretch of green. You also see some STR detectors, the CT, 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 where you see the blue and red alternating. And there's also a blue and brown. So those are the short tandem repeat detectors that we've spoken of earlier. And then there are a lot of things in there that aren't so easy to interpret, and that may be a sort of distributed representation of something we already know, or may just be features that we haven't learned are important, that the neural net has been able to discover in the data set we gave it are informative about whether a variant is an artifact or not. And this is a little bit of a tangent, but I sort of feel like everyone at the Broad should read Octavia Butler, and maybe this should just be our employee handbook, because she has this incredible series called the Xenogenesis Trilogy, which is about an alien species that comes to a post-apocalyptic Earth to basically perform CRISPR experiments on us. And one of the members of this species, they have 3 sexes. One is called Uloi, and they have these 2 arms, which are sort of normal muscular arms, and then these 2 other arms, one of which you can see at the far left, which is like a flower opening. And that's essentially like a CRISPR experimentation arm, and it can grab any biological tissue and play with its DNA. So these aliens have incredible powers. They can also sort of link their neural nets together. And at one point, a human goes into this neural net, this linked neural net, and finds it one of the worst experiences of her life. It was too intense, too fast, too alien. And I think sometimes when we're looking at these features inside the neural net, we have to keep that in mind, that this is a different kind of intelligence, and it may not always be interpretable to us. All right, great. We're doing really great on time. This is the last thing I'm going to talk about, which is that so far we've described these neural nets that build on layers of abstraction and very much go from low-level to high-level representations. We start with these edge detectors, we learn textures, then we learn parts, and finally we have an object. And we have this like very few neurons at the very end that tell us what the thing is. That's what classification is. It tells me what. But that is not the only interesting question. And in fact, there's a tremendous amount that we lose by asking only what. Another question that's also valid and informative is where. Where is the thing? And when you're answering the question what, you're throwing away tons of data that could help you answer the question where. Localize it. Segment it. Sometimes that's called segmentation. That is where you're not just saying, oh, this is an image of a cat. You're saying the cat is over here, and its ear is here, and its ear is there. And that's very useful information, too. And in a way, that is generative. because when you segment an image, you have to create another image that goes right on top of the image that came in, which tells you for each pixel what class it belongs to. So it's a generative architecture. When we're talking about classification, we're talking about compression, going from this big representation in an image to this small representation, which is just a word, which just says cat or dog or good variant, bad variant. But that's not the whole story. It's not just about compression. Sometimes we want to generate. And as always, we are stealing from evolution, because this division is at least hypothesized to already exist in our brain. So we have this visual cortex, which feeds to two different pathways. One is dorsal. One is dorsal, and that maps the visual field, and it's mostly unconscious, and it's sort of about negotiating your own motions with the visual field. The other is the what pathway, and that's sort of recognizing faces, telling you what things are, and situating everything in a global frame of reference. The where is much more local and specific to you, egocentric, versus the what, which is allocentric. And there's some of the support for this theory is in these really strange diseases that people have where, for example, prosopagnosia, where you have face blindness, you can't recognize faces, or hemispatial neglect, where you can only see half of the visual field, or simultaneagnosia, where you can only focus on one thing in the image at any one time. So I'd say there's a phone here, but there's no computer. And then someone could say, no, look at the computer, and I'd see the computer, but the phone would disappear. So all of this is, and these pathologies have to do with damage to very specific locations in the brain. There's also some support for this two streams in processing auditory information. But how does this relate to variant calling? Well, we can use this same exact idea to segment. Now we're not segmenting the visual field, we're segmenting the genome. So the genome is this 1D strip, and we can segment it into parts that are homozygous reference, that is, and little parts that are SNPs, and then bigger parts that are insertions and deletions. So we can cast the variant calling problem as a 1D segmentation, semantic segmentation problem. And then we can use an architecture similar to the one shown, or we can use a different architecture than the classification. So in classification, remember, we built up sort of like a funnel to this high-level, very abstract representation. Here, we don't do that, or we don't only do that. We build up to a high level, but then we go back down to the low level, and you see these skip connections, those curvy black lines that connect from early layers to late layers. That gives this low-level representation to the very late layers in the neural net. And crucially, there's no flatten. So we maintain that 1D structure throughout our whole processing. And at the end, we emit a variant call. So now we're not filtering a variant. We don't need variants from some truth set in order to do this. We just look at aligned reads, and then we can draw a filtered boundary. So this is an architecture which we are just sort of starting to play with, but it's an exciting way, and it's generative rather than compressive, and it's sort of inspired by this two streams hypothesis of how the brain processes images and answers for us both the question of what is the thing and the question, where is the thing? And with that, I will say big thank you to everyone, and you get to see our Halloween costume of the group Palantir. We were a bacon and tomato and arugula and avocado sandwich. And we won the 8th floor Halloween costume challenge. It's a great day. Yeah. Happy to take any questions.
Anyone has a question, just raise your hand and I'll come running up with the mic. Carrie. There you go.
I saw your comparison to deep variant, and I'd have heard of that before. I heard it's faster than other tools available for doing variant calling. Is that true? Is our tool slower? Is it?
Yeah, I think.
Are we better enough to overcome that? Like, I'm just curious. Like, what is the perception of these two tools?
I think deep variant's still, we're still sort of taking it for test spins. It is probably faster than running our whole pipeline, the way we run it. There are sort of critical differences in how it's designed. So it's like only a single sample pipeline, so they don't really have a joint calling. So it's not always a one-to-one comparison. But if you are just talking about like running haplotype caller, single sample, versus running deep variant single sample, it's probably faster to do deep variant.
Hi, I think you kind of glossed over this pretty quickly, but other than looking for the types of SNPs and deletion and insertion pattern that we know of, can you go over a little bit of the kind of patterns where maybe we didn't know before, where some new features of the genome that we're actually able to find by training your neural network?
Well, so we're doing this whole framework is all supervised learning. So it's predicated on having some truth data. So we're not going to discover new kinds of variation in this method where it's all supervised, because the only things the neural net knows about are SNPs and indels and homozygous reference. So nothing new could come out in this way. You could imagine an unsupervised structure where you would sort of see SNPs emerge and indels emerge and maybe some other kinds of maybe then it would pick up on copy number variations or structural variation as their own clusters. But that's not really available for this neural net where it's just purely supervised learning. So we are giving it this little playground to work in, but we're not allowing it to find some new classification. But within that, it may find features that we haven't thought about that are useful. And so we're sort of starting to explore that by visualizing the inside of the neural net. And we recognize some of the things like STR detectors and homopolymer detectors, but there may be other things in there that are picking up on certain very specific features that maybe we should come up with names for. But yeah, still ongoing work.
Hi, so I'm a biologist, so this might be a naive question, but in thinking about how the brain works, the different circuits talk to each other, so like the dorsal and ventral system and the two streams hypothesis would talk to each other. So thinking about like, is there a way like in computer science that you can build in that same kind of crosstalk and like switching points between like convolutional or like the generational models versus the compressive models? Totally,
yeah, and that's a little bit what's happening in this architecture with the skip connections, because we're seeing you sort of build up a high-level representation, but then you also bypass it, and then you mix the two together so they connect. But all of the architectures I presented are feed-forward. So they start with some input and go to some output. There, of course, the brain and as well artificial neural networks can be recurrent. So where we have connections not just from the input to the output, but connections that circle back. And those networks are, in general, much more powerful, but they're also much more difficult to train. So for this project, we're just sticking with feed-forward neural nets, but it is possible to design recurrent neural nets. And they do have much greater power, but they're also much more difficult to train.
Great, thank you, everybody. Have a good rest of your afternoon. Thanks, Sam.
Thank you.